And in next week, 3:10, we have the midterm exam. And midterm exam will be just like your quizzes, but mostly 50, 60 questions there. You will have two chances to do that and from home anytime. Just like that one, 3, 4 days, you will get 3, 4 days time to finish that one. Probably I will include up to translation. That is the next chapter. If I cannot finish the translation fully, then up to transcription. Okay, that will be the syllabus, the whole thing, from beginning to this one. Okay, so what is transcription? Just a little bit. What is transcription? Tell me something. Difference between replication and transcription. What is transcription and what is replication? Yes.
Transcription is the synthesis from RNA to DNA.
Transcription synthesis from RNA to DNA or DNA to RNA? The genetic, that is gene synthesis or gene expression, genetic expression is DNA, RNA, and protein. So synthesis of RNA from DNA is transcription. That is the pathway. That is the normal general pathway. And what is replication? DNA replication or RNA replication is replication. Copying the DNA strand. So what is the difference between DNA replication and transcription? Okay, the RNA is synthesized from the DNA strand, that is the transcription, and DNA is synthesized from the DNA strand, that is the replication. But what is the difference between these two? One difference is that RNA is synthesized, another difference is that DNA is synthesized. But what is the main difference here? There are a lot of differences. Make this whole process, this replication, transcription, and translation, make this figure out whole picture in your brain all the time. Okay. So in case of DNA replication, a DNA is fully replicated from endpoint to endpoint. Okay, the full DNA is replicated, new synthesis of the whole DNA, whether there is heterochromatinized region, whether euchromatin region, where there is active gene, inactive gene, that doesn't matter. The whole DNA is replicated. Okay, that is the first thing. In case of transcription, only one gene or couple of genes are producing or synthesizing mRNA. This is main part. Now the thing is that in case of DNA replication, DNA is replicated only one time in a cell cycle. Okay, during interphase, DNA is replicated only one time in the cell cycle. In case of transcription, transcription can occur many more times from the single gene. Okay. When it is required, when the protein is required, it will produce this thing. These are the main things. Next, another thing is that in case of DNA replication, one RNA primer is required. Enzymes are different, DNA polymerases. And in case of transcription, RNA primer is not required because already RNA is producing here. There is no requirement for RNA primer. And here the enzyme is RNA polymerase. Okay, now we have some kind of naming conventions here. Suppose this is DNA template. In case of DNA replication, there are two strands. One is called the leading strand, one is called lagging strand. We are naming those strands like this in case of replication. In case of transcription, there are DNA two strands are there. One is called template strand, another is called non-template strand. And what you knew that only from the template strand RNA is produced. Okay? Only one part of the DNA, only one strand of the DNA is called the non-template strand. So in case of replication, there is leading strand and lagging strand. Here is template and non-template. The functionality is different. There. Clear? These two parts. These are some kind of juggling words, but you have to remember in that way. Same DNA strand, but in case of replication, we are telling them as leading and lagging strand, and in case of transcription, we are telling them as template strand, non-template strand. Now, when the DNA is synthesized here, at that time, it is not synthesizing from anywhere. This is the gene. So what is gene? Gene means a part of DNA which can produce or which can synthesize one mRNA. Until this, what we know up to this. Now, the gene means the whole part is not synthesizing the RNA. There are some part which is called the promoter. Some operator region is there sometimes. Then the RNA coding part, this is the RNA coding part here, and the terminator sequence. So this is promoter, this is the RNA coding region, this is terminator sequence. The whole thing is called a gene. Okay? Some of the coding parts, some of the non-coding parts, their functionality are different everywhere. Now, how can I name these parts? We have to think about all the nucleotides. Okay? The first nucleotide, suppose this one is the first nucleotide from where the mRNA is synthesized. That is the first one. This one is called +1 nucleotide. Okay? So the first nucleotide from where the mRNA is synthesized, that is called the +1. There is no zero here. So if there is no zero, if I can move one nucleotide this side, that will be the -1. One nucleotide from that side, from the starting point. That is the -1. Now, based on that one, if I will go this way, all the minuses. If I will go this way, all the pluses. So this is our main part. Clear? If it is saying the minus sequences, that is called the upstream. If it is the plus sequences, then it is called the downstream. When we will say the upstream, upstream means this side. Downstream means that side. Okay, these are the naming conventions. So suppose here the example is this +34. +34 means it should be here somewhere. -75, that means here in this way. Using this naming convention, we can identify the part of the DNA. Now, just like the DNA replication, there are three main points: initiation, elongation, and termination. And initiation is the first stage of transcription. Main function here, the main enzyme is called RNA polymerase. Okay. Rather, there we had the DNA polymerase in case of DNA replication. Here, the RNA polymerase is the main enzyme, and the initiation part is that RNA polymerase needs to sit here in the promoter region. And there are a lot of functions here to put this RNA polymerase into the promoter sequence. That is the initiation. Take the RNA polymerase and put it in the proper promoter sequence, then the initiation part is done. If the RNA polymerase cannot sit on the promoter sequence, if the promoter sequence is some kind of, there is some mutation there, or promoter sequence is disbalanced, then there will be no transcription at all. There will be no expression of that gene. So that's why promoter sequence is very, very important. So this is the first part. This part is called initiation. Put the promoter onto the, put the RNA polymerase onto the promoter sequence. Then what is elongation? Elongation means in case of DNA, we know that the nucleotides are DNA nucleotides, deoxyribonucleotides, and in case of RNA, they are ribonucleotides. So addition of ribonucleotides into different the previous nucleotides. Okay. So addition of ribonucleotides. Now ribonucleotides. Now the question is that where is the position to add the new nucleotide? If you think about the nucleotide structures are similar in case of DNA and RNA. Only the position problem is that there is deoxyribose sugar, here is the ribose. So which part of the nucleotide nucleotide will attach or add? Which area? And there are two terminals, generally, two positions. One is the phosphate position, and another is that is the PO3 phosphate position, and another is the OH. This is the 5' position, and this is 3' position. Where will it be added? The new nucleotide? New nucleotide. Where will it be added? Which site?
Phosphorus.
The phosphate? Yes? Yes? There is one previous nucleotide here. Here is the phosphate group, and here is the OH group. Where the nucleotide will act? This is another new nucleotide. Where will be the bonding? These are the very common things you need to remember all the times. And if you can remember these things, you need not to think about the direction of synthesis. There will be a lot of questions like that. What is the direction of new RNA synthesis? What is the direction of new DNA synthesis? Then you have to scratch your head a lot of times. What is 3', 5', 3', something like that. But if you know this thing, you need not to think about that.
The two nucleotides hydrogen bonding.
Hydrogen bonding, that is the phosphodiester bonding. The new nucleotide addition means phosphodiester bonding. Because new nucleotide, they contains here the phosphate group, 3 phosphate groups are here. So PO like this, 3 phosphate groups, it will be added and 2 phosphate will come out as pyrophosphate, and this phosphate will add to this nucleotide. That is the thing. That is the main phenomenon. But in which side? That is it. Already in case of DNA, we had this problem because DNA polymerase cannot add any nucleotide if there is no such open that group. That's why RNA is required, RNA prime. That was the question. OH group, always the addition is here. So 3' position, always there will be the addition. We always need 3' open OH group. Otherwise, there will be no addition of nucleotide. Okay? That is the elongation. And the termination is the last stage. And main function is the release of the newly synthesized mRNA and RNA polymerase. This newly synthesized mRNA, we can call it as pre-mRNA. This is called either pre-mRNA or nascent mRNA. Because after the formation of this mRNA, it needs to go for another modification to become matured mRNA. Okay? But up to the release of pre-mRNA or the new mRNA, that is the end point of transcription. Okay. Now, what is the initiation part? The main part is the adding of, we need the substrates, that is the RNTP, ribonucleotide phosphates, and it is present in the cytosol as ribonucleotide phosphates. And we need 2 enzymes. Main enzyme is called the RNA polymerase, but RNA polymerase has two parts. One part consists of this thing, two copies of alpha subunit, single copy of beta subunit, single copy of beta prime subunit, and one stabilizing unit, that is omega. This is small omega sign. Okay. So two alpha, beta, beta prime, and omega. As a whole, this thing, this is one subunit. This is called the that is core enzyme. This is called core enzyme. So core enzyme consists of these parts, these subunits. Okay. Another factor is required, that is called the sigma factor. Sigma factor is another protein. This sigma factor is required to put this coenzyme into proper promoter region. When the sigma factor is attached to this one, this coenzyme, that is called holoenzyme. Okay. So the holoenzyme means coenzyme plus sigma factor, and that is the structure of this one. So we have two alpha, beta, beta prime, and omega. This is the actual coenzyme, RNA polymerase, coenzyme of RNA polymerase. And when the sigma factor is attached to it, that is called the holoenzyme. Okay. But in case of eukaryotes, there are a lot of RNA polymerases, and generally RNA polymerase 2, that is the main RNA polymerase in case of transcription, and this is in all eukaryotes because there are a lot of RNAs. One is mRNA, snRNA, snORNA, miRNA, there are a lot of RNAs. For each and every kind of RNAs, there are some kind of RNA polymerases there. These two are only for plants. They are not available in animals. These are only plant RNA polymerases. So in case of plant, we have 5 RNA polymerases. In case of animals, we have 3 RNA polymerases. Okay. Now what is the direction of synthesis? Because 5' position is there already, and the new nucleotide is adding to the 3' position. So new mRNA will be in the 5', 3' direction. Again, just like the DNA synthesis. It is always in the 5', 3' direction because we need the 3' position open all the time. And the new addition will be in the 3' position. So it will be in the tail. Okay. Now, the thing is that they don't need any kind of primer. So just what is required, that unwinding of the DNA double helix, and from the template strand, the RNA synthesis can occur. And from there, it is the bubble is moving, a little bit of bubble, some part of the unwinding part of the DNA, not the whole DNA needs to be unwind, just a single part, and that will move towards the DNA strands, and new RNA is synthesized in that way. Okay. Now, to unwind this part, some kind of helicase function is required. Only for this part. Okay. I'll tell you that one later. Now think about the promoter region. Promoter region must have a particular sequence. Each and every part of the DNA cannot be a promoter. Okay, promoter region can have some kind of sequences. And these sequences are called consensus sequences. Consensus sequences means some kind of common structures are there in all the promoters. Those are called consensus sequences. Mainly there are two parts. One is -10 consensus and another is called -35 consensus. So -10 consensus means from this part is the -10 region, there is some part that is called the -10. And -35 means here, that is the -35 region. Okay, there are two promoters here. This, in case of bacteria, this is called a box. And the main sequence for box is that Tata sequence. It is called the Tata sequence or Tata box sometimes. In case of eukaryote, it is purely Tata box. Okay, and minus 35 sequence, the consensus sequence is like this: TTGACA. Okay, now talk about that consensus sequences. So these are the things here. This is the coding sequence of the gene from where the mRNA is produced. Some other non-coding part is there, which is not producing any kind of mRNA, but the RNA is binding to some other part here that is called the promoter region. Okay. Now, this UTR means, this 5' UTR means untranscribed region. It is there, we don't know why, but that is not synthesizing, that is not taking part in the RNA synthesis anyway. Now, what is called consensus sequences? So, we figured out a lot of prokaryotes. This is for prokaryotes or bacteria. So, we figured out a lot of promoter sequences of prokaryotes, different species. Among these all different species, we have found that the first two codes are always T and T. Okay. The next one is mostly G. Mostly T, the third one. In case of fourth one, this is either A or C or T. Okay. So from there, we are coding in that way, the mostly T, T, G, and here is mostly A, and this one is mostly C, this one is mostly A, and this one is T. So mostly, that is called the consensus sequences. Okay. Something is common, very common, but some of the nucleotides, they are not common. In such case, we are just making it as whether it is purine or pyrimidine or something like that. So if it is purine or it is some kind of pyrimidine structure, then put that one. That is called the consensus sequences. So minus structure, then put that one. That is called the consensus sequences. So minus the transcribed region. If the question is something like that, what is minus 35 sequences or what is minus 35 promoter? The promoter region, which is situated upstream of the transcribed region, minus 35 nucleotide back, that is called the minus 35 sequence. Okay. And what is consensus sequence? Similar sequences are there. Similar sequences, not the same. Similar sequences are there, and that is called the consensus sequences. And it is mostly common in almost all the species. Generally, the protein coding part usually begins with A, T, and G. These three nucleotides, A, T, and G. Okay. And there is another part where there is no synthesis or no transcription, that is called the untranslated region or untranscribed region in case of transcription, 5' UTR. We don't know why, what is the function of that one. And this is another calculation of finding out the consensus sequences. So if T and A is here, that is fine. If Y is there, any kind of pyrimidine, that is the Y, any kind of pyrimidine, that is there. So either C or T, whatever it is. So if it is R, that means any kind of purines are there. Either G or A, whatever it is. And N means no particular base. And here mainly the cytosine and guanine is mostly there. It is GC rich region. So if the pattern is something like that, we are finding out all the species and we are finding out the first part will be like T, A rich, then there should be any kind of pyrimidines, there should be any kind of purines. If that is the sequence, that is the promoter sequence. Clear? So we have two promoter sequences here. One is minus 35 and minus 10. Okay. What is this? What is the answer to this? Question is that RNA polymer is made up of two parts. One is called coenzyme, another is called holoenzyme. Holoenzyme is equal to coenzyme plus sigma factor. Function of sigma factor is that to put the coenzyme onto the particular promoter sequence. Okay. If we know this information, then what is the answer of this one? Only the sigma factor, only the coenzyme, or the holoenzyme? A. Yes. Okay. So initiation, in case of initiation, no RNA primer is required, and location of consensus sequence determines the position of start site. Where is that one? It could be minus 35, could be minus 10, that doesn't matter. And RNA elongation, the whole part, whole RNA synthesis is done by RNA polymerase, and that is the function of RNA coenzyme. Okay. So what is the function of sigma factor? The function of sigma factor is like this. Coenzyme RNA polymerase is there. Sigma factor actually finds out the promoter region first, but there are, in case of eukaryote, there are a lot of other proteins are there which can determine the promoter site and then is to bind onto that site first. But in case of bacteria, that function is done by sigma factor. So function of sigma factor is that to find out the promoter sequence, then bring the coenzyme and put that onto the promoter sequence. That is the function of sigma factor. Bring that coenzyme, put it onto the promoter sequence, then open it up. It has the RNA polymerase itself has some kind of helicase function there. So it can open up the DNA double helix a little bit, something like 10 sequences or 12 sequences long, something like that. It can open up that one, and when it will move to the other side, they will anneal again. Okay, and no such kind of that is single strand binding protein or those proteins are not required because it is open for very short time. Okay, so already sigma factor is there. Sigma factor put the RNA polymerase onto the promoter sequence. Then the first transcribing part is open a little bit. Still the sigma factor is there. Now the first nucleotide or couple of nucleotides are added to the growing chain. When the synthesis is almost like for 10 to 12 nucleotides, after that sigma factor released. That is the function of sigma factor is done. So the function of sigma factor is that finding out the promoter sequence, put the whole coenzyme onto that promoter sequence, and start the synthesis of mRNA a little bit, and then the function is done. Clear? This is the function of sigma factor. Now the elongation is simple. It produces this kind of RNA bubble that is called the elongation bubble or transcription bubble, something like that. This bubble moves, and when it is moving up to with the DNA strand, then it opens up the next nucleotides, and the later part where the transcription is done, it is annealing by itself. We really don't know what are the proteins required for these kind of things, but we think that this is annealing by itself. Okay, and the synthesis is going on from the 5' to 3' side, and this is the whole RNA polymerase, and it is doing that function. So the transcription bubble is that just a little part on the DNA where transcription is going on, and that is little bit unwinding part. Okay, and RNA polymerase is creating that transcription bubble. Transcription bubble moves along the DNA strand up to the termination site. Addition of nucleotides in that bubble the nucleotides, new nucleotides, they are already present on cytoplasm as a ribonucleotide phosphate. Those ribonucleotide phosphates are coming to that bubble and they are added to the previous nucleotide at the 3' position and they are forming the hydrogen bond between the DNA template and the RNA template. It is for sometimes then the RNA is released from there. These hydrogen bondings are cut out and the RNA is released. Now, based on that one, what I was telling last day also, that if you think about the RNA part, that is the nucleotide sequence of RNA, that is the complementary of the 3', 5' or the template strand of DNA, right? It will be always complementary. So if G is there, here will be C. If A is there, here will be U, because in RNA there is no T, always U. If C is there, here will be G, always complementary, all the sequences. Now this sequence is complementary to this one in the DNA. These are DNA double strands. This sequence is complementary to this one. Definitely, should be. So here G is there, C is there, A is here, so T is there, like this. Now, what is the similarity between these two? RNA and the non-template strand. Is there any similarity? So basically, if we know the sequence of non-template strand of the DNA, we can have the sequence of the whole mRNA. Okay, and this is required for our bioinformatics study when we are doing the sequence of mRNA. And sequence of mRNA and studying mRNA, that is called transcriptomics. mRNA is called transcriptome and the whole subject is called transcriptomics. So this is the elongation process. Elongation is very simple, just adding the ribonucleotides onto the wedge part of the previous nucleotides and it is going on. That. But the termination part, there are two main factors here. One is called the row-dependent terminations. There is some protein called the row factor. Row factor is required for the row-dependent termination. And row-independent termination, where the hairpiece structure is formed and followed by a string of uracils and a terminator sequence is there. No row factor is required. So what is the row-dependent termination? Row factor is another protein and it is present near the transcription sites. So RNA polymerase is moving towards the DNA strand and it is forming the new mRNA synthesis, new mRNA chain here. But when RNA polymerase that is reach the terminator sites, terminator site means there are some kind of sequences there and which is called the terminator sequence. If it reaches the terminator site, then it halts the synthesis. Okay? And when it halts the synthesis, at that time, row factor binds to the new mRNA chain. So binding of row factor is not onto the DNA chain. Binding of row factor is onto the new mRNA chain. That is main part here. Now, when it halts, at that time, it produce some kind of hairpin structures. Because in terminator site, there are some inverted repeats. Inverted repeats means something like that. So, here is AACG there. Now, there are some kind of codes here. After those codes, we have C, G, T, T. So these are called inverted repeats. So in such case, A will pair with this T, this A will pair with this T, C will pair with this G, and G will pair with this C. So it will form a hairpin structure. If you just move in this way, it will form the hairpin structure. So that is the way this hairpin structure forms for these inverted repeats. Okay. And this is the main part in the terminator sequence. There are some inverted repeats there. So as a result, when the hairpin structure forms, that means it is indicating the synthesis is done. RNA polymerase stops here. Synthesis, pre-mRNA synthesis is done. Rho factor is moving to this new mRNA chain. Then it gets that RNA polymerase there. When both of them are in contact, that rho factor and RNA polymerase, they release. They dissociates from the DNA chain. Okay, and that is the termination. So this is one way called the rho-dependent termination. But if rho factor is not there, what is happening? The rho-independent termination is that there is some inverted repeats. That is the terminator sequence. That is the part. Here the sequence is produced also. And after these inverted repeats, there are a lot of repetitive sequences, like a lot of A. RNA polymerase cannot go to lot of RNA A sequences. So we can say that it became bored. Okay, I cannot synthesize anymore. That same thing. It dissociates from there. Okay, that is called the rho independent termination. But that doesn't mean that each and every mRNA should have that kind of A there at the end. A means that is forming the U. But generally, each and every mRNA contains two main parts. One is called the methyl group on the top, on the front part. This is the mRNA, and at the end, this is called the poly A tail. All the A's are here. A lot of it. And this is actually the synthesized mRNA. Each and every mRNA has these things. I will talk about this one later. But that is not from this termination site. Okay. In case of eukaryote are higher organism, they have a lot of more functions there. As that is quite simple. They have a lot of more proteins there, a lot of more enzymes there, and their function is a little bit different. And main thing is that they have chromatin. Chromatin means histone proteins. In case of bacterial DNA, there is no such kind of histone proteins. But in case of eukaryotic DNA, there is histone protein. So histone protein is there, and here the promoter sequences have a lot of things. One is called the basal transcription apparatus. Transcription activator proteins are required. RNA polymerase. There are a lot of RNA polymerases, but the main RNA polymerase is RNA polymerase 2 here. And the Tata box, the promoter sequence. But here the part is different. Here it is -25 to -30 region. In case of bacterial promoter sequence, it was -15, -35, and here is -25 to -30. So chromatin structure modification, we will talk about this one later in our eukaryotic modification chapter, that is after the bacterial one. Okay. Chromatin have different kind of things. They have, we knew already that one, that each and every histone has some kind of protrusion. So this is the histone protein, histone octamer. They have some kind of protrusions like this. Okay. And these protrusions, they are there to anchor the DNA. They needs to be released a little bit less. So rather than this side, comes to this side. So they can loosen up the DNA. This is called the histone modification. They can loosen up the DNA. As a whole, only for the slight part of the DNA, the replication bubble can form. That is the transcription bubble can form. And there are some other things, that is main enzyme is called the acetyl transferase. They add acetyl groups to amino acids at the end of histone proteins and which destabilize the nucleosome structure. Addition of methyl groups, they also destabilize the chromatin packing, the tight packing. And chromatin remodeling proteins may bind to the chromatin and displace nucleosomes. This is one thing, that is DNA was coiled 1 and 3/4 times here. Okay, it needs to be open up. Anyway, this part of the DNA needs to be open up because we don't know that the always the DNA, that is the transcription part, is within the linker DNA. It can be within this part, so it should be opened up. That one part is there to open up these anchors. That is the function of acetyl transferase, and displace this nucleosome from here to open up the whole DNA. So it should be dissociated for a little bit time. To open up this whole DNA, then the transcription is done, it can come here again and can bind again. So these are the functions between, this is the difference between eukaryotic DNA and prokaryotic DNA. Eukaryotic DNA is packed on the histones, so that needs to be unwind anyway. And these are the functions to unwind that part. So we have three different kinds of eukaryotic RNA polymerases, polymerase 1, 2, 3, and they have different promoter regions because they are not responsible for the all nuclear DNA, and they are responsible for mitochondrial DNA, some kind of chloroplast DNA, or some other things. So they have different kind of promoter sequences. And a lot of accessory proteins are required to find out the promoter sequence. Not, there is no sigma factor there. RNA coenzyme is there, but there is no sigma factor there. Instead of sigma factor, there are a lot of other proteins are there, which is very, very required to use to put this RNA polymerase coenzyme onto the proper place, onto the particular promoter region. So one is called the transcription factor. This is a class of accessory proteins which binds to the DNA sequence to find out the promoter region and then attach the RNA polymerase. And general transcription factors, they bind to the RNA polymerase to take it to the actual promoter region. And these are the group of proteins that assembles near the transcription start site and start the transcription, initiate the transcription. So one is binding to the DNA, some proteins. General transcription factors, they are binding to the RNA polymerase, and this communication between these two proteins, they are required to attach the RNA coenzyme onto the particular promoter site. Not only that, there are some other things and some other regions are there which is very, very required for this kind of bindings, and there are a lot of other proteins. I'll tell that one. So RNA polymerase 2 is the main enzyme. In case of eukaryotic protein, we can say transcription factor 2 is transcription factor for RNA polymerase 2, and that is P. So transcription factor 2A, transcription factor 2B, that means two different proteins. Both are transcription factors and both are required for RNA polymerase 2. If transcription factor is required for RNA polymerase 1, then we can say TF1. If there are a lot of proteins there, A, B, C, D, whatever it is, then we can mark them as ABCD. Okay. So there are a lot of promoter site, and this is the TFB recognition factor site where only the TFB proteins will be attached. This is the main promoter sequence, that is the Tata box, where the RNA polymerase will attach. These are the initiator element, all other proteins which is required for initiating the transcription. This part is responsible for that one. And this is downstream core promoter element. So the movement of the promoter to the downstream, it is recognizing that. And that is actually mainly the transcription site here.